Okay, so why did I color in some of the chromosomes as red and blue? I could have colored in these two, the other two cells, but I only did this one and this one. What do the red and the blue represent? Yeah. Nope. Good guess, though. Mom and dad, right? So the red chromosomes are the ones that you would get from your mother, so they're the maternal chromosomes, and the blue are the paternal chromosomes, okay? So, if we look at these chromosomes, this would be both red, right? So the question about, right? So this would be red, right? And that one would be red, right? And then these would be red, right? So the A chromosomes are red and the E chromosomes are blue. Okay, so are there, is that first cell, is it haploid or diploid? Haploid, right? Because it only has half of the genetic material. So remember that you always get a one copy of the chromosomes from one parent and the other copy of the chromosomes from the other parent. So that would be a haploid cell, okay? So what is the other question? So number 2, are the chromosomes here, are they replicated or unreplicated? They are replicated, right? So the chromosomes have made a copy. So when we look at this, let's look at these two first. So are these homologous chromosomes?
What's homologous?
Homologous means the same, they have the same shape, the same size, they have same banding, staining problems, same genes, right? So these are not homologous. So here we have homologous chromosomes, but they're replicated. So we would say that we have pairs of replicated homologous chromosomes.
Is that the sister chromosomes, or is that different?
That is different. Oops, what happened? Oh, moved it. Technological difficulties. So these are pairs. I'll explain sister chromatids in a minute. Okay, so that should say pairs of replicated chromosomes. Right. So the sister chromatids are right here. So those would represent the sister chromatids. So these are sister chromatids. Okay. And the sister chromatids get pulled apart during mitosis and meiosis. So this could be a cell that is ready to reproduce either mitotically or meiotically, right? So it could be either going into mitosis or going into meiosis. So we don't know. So that first stage is the same, okay? So those are replicated chromosomes. So is this cell haploid or diploid down here? It's not haploid, it is diploid, right? So the reason why it's diploid is that there's a copy of there's a copy of the genes, right? So if I was to write the genotype, it would look like this. Right, so this should look familiar to you from the lab, right? So this is a diploid cell. It has two copies of the A gene. It just happens to have one dominant A and one recessive A, but there's still the A gene, right? E is on a separate chromosome, and we have heterozygous. So this would be a heterozygous for the A gene and the heterozygous for the E gene, okay? Okay. So that is the genotype. Would this be a gamete or a body cell, a somatic cell? What do you think? It would not be a gamete. Seeing it. So gametes are haploid. Somatic cells are body cells. So this would be like a skin cell, a liver cell, a pancreas cell, a neuron, right? This would be a body cell, so a somatic cell. So the gametes are the ones that have only half of the genetic material, right? Right, have half of the genetic material. Okay, so this one is actually a little bit harder question. Do we have, these chromosomes are replicated, are there pairs, are there homologous pairs of replicated chromosomes? I'll give you a midterm back at the end. Okay, here's the homework.
I'll go with yes.
No, okay. Okay. There's sister chromatids, but is there, I said that this was where there are replicated, right? There's homologous pairs, because these are the homologous pairs right here, right? This is just a single replicated chromosome. It is not a pair.
So if there are pairs to be both A's.
Yes, so if they were a pair, they would be both A's. Excellent. But those are sister chromatids. Those are not pairs. Right? So this is a pair of unreplicated chromosomes right here. So pay attention. Okay, we want you to get this. So this is pairs, right? This is a pair and this is a pair, right? They're unreplicated. These are not pairs because they have different genes, but they are replicated chromosomes, okay? So the harder question is this at the end of meiosis 1 or meiosis 2? We'll get to it in a second. Meiosis 1 or meiosis 2? 2, oh, thank God, okay. It is meiosis 2, right? Because at the end of meiosis, actually, oh God, it's meiosis 1. Oh no, I'm serious. Okay, the reason why it's not meiosis 2 is that the chromosomes are still replicated. So in meiosis 2, we actually separate the replicated chromosomes. So at the end of meiosis 2, we would have a gamete. This is not yet the gamete. This would be the end of meiosis 1. Okay, Madison?
So the cell on the left, so are those homologous pairs as well?
Yes.
Okay.
They're unreplicated. So you could write homologous pairs of unreplicated chromosomes. Right. Those are pairs. Okay. So you're asking yourself, why would I even care about this? Right. Well, the reason is this is essentially why, you know, the way that our Mendelian genetics problems, the way that they work, right? So we're going to talk about what would be the possible combination of genes that you could then get into a gamete. So this is a genotype, right? So when we do our, we're going to look at some crosses where we try to figure out, well, a gamete would be this, right? And that means that I have one copy of the A chromosome and I have one copy of the E chromosome, right? Right? And so we're going to talk about the inheritance of two traits, A and E, or some other combination of letters, right? And we're going to talk about thinking about how the chromosomes behave during meiosis, and then how they would subsequently come back together to form offspring. Okay. But before we do that, we're going to talk about the quiz next week. Okay. So we're having a quiz next week on Thursday. And it is going to be over Mendelian genetics. So it's over kind of what we did in lab, and the lecture that you were supposed to watch. If you haven't watched it, you want to watch it. And then what we're going to cover today. Now, I might, we'll see how far we get today. I might do another recording. Okay, so I will let you know at the end of today if I need to do another recording or not. Okay, so you're asking, well, how could you do a multiple choice question off of those questions that we looked at in lab? So this is it, okay? So let's look at this example. So this is a disease that we already saw. It is autosomal dominant, okay? So notice how it does not skip generations. when we figure out the generations, remember that the generations are a Roman numeral. So we have 1, 2, and 3, okay? Then within that generation, we number the individuals, 1, 2, 1, 2, 3, 4, 1, and 2, okay? Then if I was doing this problem, I would do my legend, right? So big F is what? The disease. And I know that because it's autosomal dominant. So little f is normal, right? So all of those individuals on this pedigree that's family history that are not colored in would have to have what genotype?
C.
Little f, right? So I could fill those in, little f, 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 okay? And so I look at what individual am I talking about? It's actually 2, 1, right? And so that would be that individual right there. And so the correct answer is C, okay? A harder question would be, what is the genotype of individual 1, 1?
That's right. Big F.
Okay, well, you have to look at their offspring. Okay, so they had an offspring that had a little f. So it means that they had to get 1 little f from the mother and 1 little f from the father. And so this father would have to be heterozygote, right? So big F, little f. Right? Does everybody see why? Working backwards. Taking into consideration all the information in the pedigree. And everybody actually that has this color name would have to be big F, little f. There's no way for them to be big F, big F. And that's actually true for bad genetic diseases. It'd be very rare for them to get 2 dominant copies. It would have to be 2 individuals with the same mutation having a child that had both bad copies of the gene. Okay, any questions about this? Okay. So before we continue with Mendelian genetics, I want to talk a little bit about how we can get errors in meiosis that lead to chromosomal abnormalities in number and in the way that the chromosomes look. And so when we look at meiosis, we can have what is called non-disjunction. And this is where the chromosomes do not separate properly during meiosis. So during the formation of the egg and sperm. Now, if they do not separate normally, this can lead to a phenomena that is called aneuploid E. Right? So if you break this down, this word, A-N, this means without. They just put an N in there. A always means without. They just put an N in there because there's an E here. U means good, and ploidy means copy. So this is without a good copy, right? Or a good number. Ploidy could be the number, right? So if you remember we were talking about diploid, diploid is 2N, where in humans, N is 23, right? So we have 2 copies of every chromosome, and we have 23 total different types of chromosomes, right? So aneuploidy could be 2N plus 1, or it could be 2N minus 1. If it's plus 1, this would be 46 plus 1. So that would mean that they have 47 total chromosomes. If it's minus 1, then that would be 45, right? It'd be 46 minus 1, which would be 45. Okay. So what is a trait that is due to an extra copy of a chromosome? Down syndrome, right? So we can look at Down syndrome. Does anybody know which chromosome do you have an extra copy of? 21, right? So it's actually plus 21. So it's one copy of the 21st, an extra copy of the 21st chromosome, okay? So if we look at a karyotype, which is a picture of the chromosomes, so this is your homework. Right? We can visualize the chromosomes, and if they're fetal chromosomes, they're generally obtained through amniocentesis, which is where you take a little bit of the amniotic fluid and then you separate out the fetal cells, and then you grow the fetal cells in a petri dish, and then you get them when they're undergoing mitosis, and you squish them and you stain them, and then you look at them under a microscope. So this is what a karyotype can look like, okay? So you'll notice one thing, that the chromosomes are lined up, and you'll notice that the first chromosomes are really big. But when you get down to the 22nd chromosomes, they are smaller, and then they stain in a particular way. And we'll talk about what that stain means in terms of what genes are being turned on and turned off. But notice how the banding pattern is the same. So this would be a homologous pair of chromosomes. And you're like, well, those don't look homologous. But think about it, they're being squished, right? It's a chromosomal squash, they call it. And they're getting, you know, a picture taken of them. So they could be like in weird, they could look weird, right? But people that do this for a living are like, they are super fast at this, right? They're like, oh, I can, you know, they just like memorize what chromosomes look like. And they have a search, and then you just go boom, 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 right? But us trying to figure out what the chromosomes are would be more difficult. Okay. Also notice that the center of the chromosome, the center here, is in different locations. So notice that this one has a really, this is called the P arm, and this is the Q arm. So it has a small P arm and a longer Q arm, okay? So when we look at this, is this a female or a male? Female, right? And so we would write it, we would look at, and you can see this on your handouts. We could write it as 40, your homework, you're 47, and then it's a female, so it'd be XX, and it would be plus 21, right? So I'm not going to make you match up and figure out like all of them. I did this once, and my students reported back that it took hours to do it, okay? So this should not take longer than a half an hour, okay? So what you're going to do is there's only in the program that you're going to go to the website, and the program has most of them already matched up, and so then it gives you one and says, okay, well, where is this one? And it only gives you a few options. And if you screw up, it says incorrect, try again, right? So it tells you right away if you match them up incorrectly, okay? So we're going to look at that, okay? Now, there is a special type of aneuploid, which is called a sex aneuploid. And the sex aneuploid means that it has to do with the sex chromosomes. So it's the X and the Y, right? So X or Y. So it's kind of interesting when we think about this process of non-disjunction, and we think about what embryos we see. I don't believe that this process of non-disjunction, which looks like this, so the chromosomes are not separating properly. So notice how they line up, and these two go to the opposite ends, and so we get a gamete here with 3 chromosomes instead of 2, and they actually have a pair. And then we have a gamete here, which is missing a chromosome, okay? So when you think about it, it seems to me, I believe, and I think other people believe this, that it could also happen to the first chromosome, the second chromosome, the third chromosome, but those are such huge chromosomes that if you had an extra copy or were missing a copy of that chromosome, then you would never see a baby being born with that defect because it would just be too big of a defect, right? So babies would be spontaneously aborted or miscarried early on if they had that dramatic of an effect. But if you think about the 21st chromosome, that's a pretty small chromosome, right? And so that's not a lot of extra genetic material, okay? And then if you think about the sex chromosomes, males only have one copy of the X chromosome. So we could sometimes see people being born that are XO. And this is the only instance of an aneuploid in humans where you could actually be missing a chromosome. So not an extra chromosome, but you're actually missing a chromosome. So that O means that there's no second sex chromosome. There's only one X chromosome. Now this person is female, right? They're female still, but they have some problems reproductively. They tend to be infertile. And this is what is called Turner's syndrome, right? So just to be clear here, this is lethal. You would never see a baby being born with no X chromosome. That would be lethal, okay? Now, we can see people being born that have this combination. And do you think that individual would be male or female, like phenotypically, like if you were looking at them, their genitalia, and you were looking at whether or not they had a testes or ovary, right? That is male, okay? So male symbol, there's the male. And that is due to the Y chromosome. So the Y chromosome has a gene on it that determines if an embryo is going to become a male or a female. So if it has a Y chromosome, it will always be a male, okay? So this is a male, and this is Klinefelter's syndrome. Right? Okay, so we could also have XYY. Okay, so that would obviously be a male. Right? And this is what is referred to as Jacob's. I don't know if it's called syndrome. Jacob's genotype, blah, 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 syndrome. Syndrome. I don't, it's not called syndrome because it actually is not that dramatic of an effect on the phenotype. And so in the 1960s, there was this huge deal wherein they went into a prison and they looked at the genotype of inmates, and they found that there were quite a few males that had the XYY, right? And then they made the assumption that because there were so many males, that was called a super male, right? And then they made the assumption that men with that genotype were more aggressive and more likely to end up in prison. So there was briefly a period of time when the idea was that we need to test all baby boys. And if we found out a baby boy is this, then we treat them differently than if they are XY. And that was like really bad because then you're assuming that is genetically predetermined and you're going to treat those individuals differently and possibly that could actually affect them, their phenotype, right? That could actually affect how they turn out. And so the study was actually flawed because when you look at the population as a whole, there is just as many people out there in our population, outside the prison as there is inside the prison having this genotype. So we no longer believe that it causes an increase in aggression. What it does cause is people being taller and bigger, and they tend to have bigger hands and feet, right? And so that might affect the way that they are treated, and that might affect their propensity to maybe have trouble in our society. They're big hands and big feet. Okay. So that's Jacob's. Okay. We could also have this, right? And this is a super female, right? Female, not femate, female. And so we can have people that are XXX and we don't even know it, right? There's really not much difference in their phenotype. Okay.
So does the amount of those sex chromosomes, does that have any effect on what they pass down as far as the sex gene? Like,
Yes, and so they might have a harder time, specifically this individual might have a harder time maybe producing normal sperm. Because if you think about it, if they have an extra Y chromosome, then they might pass on the 2 Y chromosomes. I mean, it would affect the genotype of their offspring. Okay, so that is aneuploid. So that is your homework. Okay, besides aneuploidy, we can have additions or deletions to, so this would be an error in meiosis as well. So this is addition or deletions to chromosomes or deletions in chromosomes. So this is error of meiosis, but it's not aneuploidy. Okay. So sometimes, if you think about crossing over, which is that rapid exchange of genetic material in prophase 1 of meiosis 1, you could think, well, maybe the enzymes would cut that DNA and then it wouldn't put it right back. And you can actually get translocations as well. So you can get pieces of chromosomes put on the wrong chromosome, right? But in this particular instance, this is a deletion in chromosome number 5. So a geneticist looking at this karyotype would see that there's like a missing band and like there's a piece of that missing. And so if we look at it in more detail, it's like this is the deleted region, okay? So that is a big mutation to lose all of that. And that ends up on chromosome number 5. That's actually a pretty big effect on the phenotype. So this is a syndrome that is called Cri-du-Chat. Right, so deletion on chromosome number 5 is called Cri-du-Chat. You want to write that down? Yeah.
So if like the Klinefelter syndrome, the XXY in a super female would have a child, does that have any effect on like whether they're having a boy or a girl with like the X's? I don't know if that's a good question. The X's like. I didn't know if it was like the capital F and then like the V.
I don't think it would. It might mean that they would have a child that would be be more likely to be XXY or XYY. I don't think it would affect the difference between male or female. Yeah, that's a good question though. Okay, so Cri-du-Chat is caused by a chromosomal deletion on chromosome number 5. And this is cry of the cat. Because one thing that this affects is their larynx. And so they have kitten-like cries rather than normal crying. And then it has other symptoms. And so this would be a person that has Cri-du-Chat, and that so it was first discovered by a person that was French, obviously, right? And so they tend to have microcephaly. So they have a larger than average head. And then they have certain phenotypical characteristics, including some mental deficiencies. And so that deletion had a big effect on, and so we see that there are a few of these examples, but if you can imagine if you deleted a large part of a chromosome, it would probably lead to such a bad defect that the embryo would not be able to survive. Maybe it wouldn't even be able to implant, right? It would just be spontaneously aborted. Okay. So there's a lots of mechanisms by which we can protect, the female protects against having babies that have really bad genetic defects. Okay, so we're gonna continue on with the Mendelian genetics, and we're gonna talk about what is referred to as a let's not talk about the dihybrid. Let's look at inheritance of 2 traits. Okay, so let's pick an example from what we did today, or what we did in lab this week. So we could do tongue rolling. So we could have R gene. Right? And then we could do, what's another one? Index finger. That's different, male or female, isn't it? Yeah, so we don't want to do the short index finger. How about the dimples? Okay, so this would be the D gene. Okay, so these are on different chromosomes. So a person could have that combination. Okay, so that means that they are heterozygous for the R gene, which they can roll their tongue, and they're homozygous dominant for the D gene, so that means they have dimples. Okay, so they roll their tongue and they have dimples. Okay, and then we can look at a genotype that would be RR, big D, little d. Okay. Now, we want to talk about the combination of alleles as they end up in the gametes. So we want to write down that these are genotypes. Those are genotypes. That would be in a dihybrid cell, right? So what we have to do is we have to figure out the combination of alleles that would end up in a gamete, which is haploid, right? Not dihybrid, diploid. So it'd be a haploid cell, okay? So combinations, we could have a big R and we can have a big D, right? So you see that big R, big D. That could be one possible combination. The other possible combination is we could have little r and little d. Okay, so those are my gametes. So just like we did at the very beginning of class today where we had A's and B's, now they're R's and D's. Okay, and then somebody told me, and I don't remember this from math, but foiling. They said that this is the foiling method. Okay, I'm not sure what that means, but I figure out R goes to D, right? That's one possible combination. So I always do it in this way. R, big D, big R, little d, little r, big D. Is this foiling? I don't know. Little r, little d. Okay, so it's all the possible combinations. So big R could end up with big D. Big R could end up with little d. Little r could end up with big D, and little r could end up with little d. Okay, so these are gametes, right? They have only one copy of each gene, okay? So I can put them together in a Punnett square. And when I do this, I always put the R's together and the D's together, just for simplicity. I don't know why I made that I made it kind of screwy. Should be equal, okay? So just for simplicity, I put the R's together and the D's together, okay? So the genotype of the offspring would be this. And I always put the dominant one first because then it is easier to read too. Let's see, big R, big D, big R, big D, little d. Little r, D, little r, D, D. Okay. Okay. So these, right, are the genotypes of the offspring, of the possible offspring. So this is my genotypes of possible offspring. Okay. Now the phenotype would be what they look like, right? And so most of them look like the parents. They have the ability to roll their tongue and they have dimples. But this one cannot roll their tongue, right? This one cannot roll their tongue, but they have dimples, right? And so did I do that right? Yeah. Okay. So these would be different from the parents. So 2 out of 8, right? Or 1, that'd be 1/4, right? Would be different from the parents. Okay, so we'll have some more practice figuring out the gametes from the genotypes. Okay, so let's talk about incomplete dominance. And so we have been assuming that alleles are either dominant or recessive. So on one end of the scale, I have what is referred to as complete dominance, right? So that would like be the ability to roll your tongue. So all those Mendelian traits that we looked at, you either could or you couldn't. There was no in between. There was no medium, right? On the other side, we have what is called co-dominance. Okay. So this would mean that the alleles are equally expressed. And then somewhere here in the middle, we have what is called incomplete dominance. Well, I have it written up there, but incomplete dominance, right? So this is like a gray area. Most of the traits that we talk about, even cystic fibrosis, is a little bit incompletely dominant, right? So we can talk about, you know, sometimes we assume that they're completely dominant when they might just be a little bit incompletely dominant, okay? So this was actually discovered first in 4 o'clocks, and it's 4 o'clocks are flowers that open in the afternoon, where we had a red flowered plant, and we crossed it with white flowers, and lo and behold, we get pink. Right? So that actually supports the blending theory, where the offspring are always intermediate between the adults or the parents, right? However, in the next generation, if we take pink flowers and pink flowers, right, we then get red, white, and pink. So they get separated back out. Okay. So this does not support the blending theory, because the blending theory says that once you put the traits together and you blend them, then you would never get them back out. You never get the variation back out. Okay. So in this case, because it's incompletely dominant, we're going to use R1 and R2. Okay. So R1 codes for red, R2 codes for white. So notice that it's the same gene, but because it's incompletely dominant, I didn't want to use a little r, okay? So red would be R1, R1, right? White is R2, R2. Pink would be heterozygous, right? This one would be heterozygous. And you could put these in a Punnett square like this, where you'd have R1, R2, R1, R2, and then you'd have red. 1/4 would be red. That's pink. This is pink. And then this one is white. Oops, R2. So 1/4 would be red, 1/2 would be pink, and 1/4 would be white. So this is, in humans, this would be any trait where if you're heterozygous, so if you have one copy of one allele and one copy of the other, if you are different in your phenotype from being homozygous dominant or homozygous recessive, then that is one that is incompletely dominant, okay? So let's look at humans. So the human disease is familial hypercholesterol esterol. Spell it right. Cholesterol. Emia. Okay. So familial, I didn't spell familial right either. Okay. So familial means family. So that means it's genetic. So anytime you see familial, that means it's genetic. So it's passed down in the family. Hyper means high cholesterol, right? So this is due to a defective receptor that, it's a gene that codes it for a defective receptor that then would prevent cholesterol by being taken out by the liver. So blood would go to the liver, but the cholesterol that you eat would not be taken out of it, okay? So I'm gonna show you just a little video that talks about this particular pattern of inheritance. Oops.
Familial hypercholesterolemia, or FH, is a genetic disorder characterized by greatly increased LDL levels and a family history of premature atherosclerosis. Normally, LDL receptors on liver cells help clear LDL particles from the bloodstream. FH may be caused by any of over 1,000 specific mutations that affect 3 known genes. More than 80% of these mutations increase LDL levels by causing the LDL receptors on hepatocytes to either be functionally altered or underexpressed, decreasing the body's ability to clear LDL from the bloodstream. The result of either scenario is an increased plasma concentration of LDL particles. In addition, impaired LDL uptake also decreases the cholesterol pool in the liver, which stimulates hepatic cholesterol synthesis and the production of VLDL, a precursor lipoprotein to LDL. These defects found in FH can result in an increased concentration of LDL particles starting early in life. Sustained exposure to high LDL levels and increased deposition of cholesterol into the artery walls over time elevate the risk of atherosclerosis and premature cardiovascular disease associated with FH, making early detection of this genetic disorder of critical importance.
Okay. So that's the phenotype. So that would be how the gene affects the body. Oops. Not. Okay. So that's how the gene affects the body at the cellular level. And then it can lead to heart attack. So let's look at this gene FH. So let's say that FH1 is normal. And FH2 is abnormal. So that's the one that leads to the disease, okay? So you could have one normal allele and one mutant allele. And then this causes heart disease when you're in your 20s to 30 years of age, right? So abnormal levels of cholesterol can be like a heart attack in a 30-year-old or 25-year-old, okay? But it is worse if you have 2 copies of the defective allele. So if you're FH2 and FH2, then you have heart disease when you are younger, right? So sometimes you hear of people when they're teenagers and they're like running around the fields at school, and then they just die of a massive heart attack. And so that generally is due to this hypercholesterolemia. So this would be like in children, you could have a heart attack. I think there was a famous basketball player that died because of a massive heart attack. due to his having hypercholesterolemia. Okay. So notice how there's a difference in if you're heterozygous or if you're homozygous for the mutant allele. So we see a difference, just like with the red and the pink flowers. Okay, so that was incomplete dominance. Now I'm going to give you an example of multiple alleles. And so the best example of this is our ABO blood types. So the gene that codes for blood types is gene I. I'm not sure why they give it an I, but that's the gene, right? And we have 3 alleles. So we have I with a big A, I with a big B, and then we have little I, okay? So these right here would be said to be co-dominant, which means that they are equally expressed. The little I is recessive. Right? So even though we have 3 different alleles in our population, we only have 2 in our body, right? So we only have a combination of 2 because that's our genotype. So if we look at the genotypes that are possible, we have this combination or this combination, okay? This produces the phenotype, which is blood type, right? So this is type A. Both of these would be type A, okay? And what that means is that you have type A proteins on the surface of your red blood cells. So if you're type A blood, you have type A proteins on your red blood cells. We talked about how there are proteins in the plasma membrane and that are on the surface that allow the cells to recognize one another. So that would be one of those, okay? You could have this combination. That is type B. You could have this combination. That is type AB. Or you could have this combination. This would be type what? O. Okay. So if you have type O blood, you know that you are little I, little I. If you have type A, B blood, you know what the genotype is. So I'm type A, and I don't know if I am big A, or if I'm big A, little I, right? So I don't know what my genotype is just by looking at my phenotype. Okay. Okay, so now we have the positive and the negative, right? This is a separate gene, and this gene is what is called the Rh gene. So you might have heard of Rh factors, right? So the Rh gene, it actually was first discovered in rhesus monkeys. And we also have it, and it's particularly important when a female is pregnant, because if she doesn't have the Rh protein, then she could produce antibodies that are going to attack the fetus, okay? So let's look at this gene. So if we are Rh positive, okay, this is dominant. And we have Rh proteins on our red blood cells. Okay. If we are Rh negative, this is recessive. So we lack Rh proteins. Okay. So this goes back to our inheritance of more than one gene. So what would a person that is this combination be? What would be the blood type of this individual? A positive, right? If I have RH negative, then it's negative, right? So what about this one? O negative, yes. Okay. Now, even though we have 3 alleles, remember we can still use a Punnett square to determine the probability of having an offspring that has a particular blood type. So if we say that these are the 2 parents, okay, and I want to figure out what is the probability of them producing a child that is O negative. Okay, so what's the probability of those 2 individuals producing a child that is O negative? My first thing that I would do is I'd figure out the gametes that each one of these could produce. Okay, so this one could produce this gamete, right? This one could produce 4 different gametes. It could be big A, RH positive, right? Big A, RH negative, little I, sorry, I'm gonna write it up here, little I, RH positive, and little r, RH negative. Okay, so these would be my gametes. Okay. And just by looking at that, how many times would we have offspring being produced that would be little I, RH negative, out of 4? Because it'd be like a 1 by 4 once, right? So we'd have to have these 2 coming together, right? Those 2 coming together. So you could do a Punnett square, which would be 1 by 4, right? And you would find that there's a 25% probability that these 2 individuals would produce an offspring that is O negative. Oops, wrong way. Okay. So this is the last thing I want to talk about, and it is called epistasis. And epistasis is where we have 2 genes interacting to determine the phenotype. Okay. So if 2 genes interact, then we have to look at the inheritance of both of them to determine one phenotype. Okay, that's totally different than what Mendel did in his experiments, right? Now, my favorite example of this is brown versus blue eye color. So we're going to completely forget what you might have heard about the inheritance of eye color. Right, completely forget about one being dominant and one being recessive, okay? And we're gonna ignore green, because green is due to another gene altogether, okay? It's due to the, so brown and blue eyes are actually due to the presence of melanin in your iris, and it's how much melanin, and then its distribution can determine the subtle variation. So we get lots of variation in eye color, right? Okay, so we have a gene, which is the O gene, right? This codes for melanin, right? So we only have one form of that, unless you're albino and then you don't have any melanin produced. But most of us have melanin, right? So we have skin color and we have hair color, right? So we have melanin. And then there's another gene that is called H. And this codes for melanin production in the eye. So it'd be the melanin distribution, or melanin presence. We'll just say melanin presence in eye, right? So in this one, we're going to say that there's 2 alleles. So H is normal. Little h means that it's abnormal and there's no melanin. And this gives you the blue eye color. Right? Okay. So when we look at the evolution of eye color, in the past, everybody was this. Right? This would produce brown eyes. Okay. However, we could have this combination, and these two genes interact to give us blue eyes. Okay, so they're interacting to give us blue eyes. But notice how we have to have 2 copies of the little h gene to get blue eyes. Okay? So if we look at this, there's been a lot of recent research about this blue eye thing. And so if we look at, you might have seen, okay, says blue-eyed humans have a single common ancestor. So what that means is that they have tracked it back, this mutation, they've traced it back, and it only appeared 6,000 to 10,000 years ago. Okay? And it was in Europe that this mutation appeared that gave people blue eyes. And so it's believed to be up until that point in time, everybody had brown eyes. Right? There were no blue-eyed humans on the planet. And so they were able to find DNA from a person that had blue eyes. And so this is kind of what they think that person might have looked like, okay? So notice how startling the blue eyes are, right? And they then have this dark skin color. This is a hunter-gatherer in Europe, right? So the question is, if this mutation only arose once, how do we have blue-eyed individuals? Right? We had to have had somebody mating with some relative, right? Could have been second, third, fourth cousin, right? But we had to have somebody mating in order to get a combination of those genes passed down into the offspring, okay? So if we look at an example of this, let's say a pedigree, let me go back, okay? So if we look at, okay, so everybody is big H, but this person, this is the single mutation. Right? That's the single mutation. And then we had people, and this is just a small pedigree, right? It could have been, I'm just going to do first cousins again. Okay. So that single mutation, way back when, right, was passed down. And then if you have individuals that have gotten that mutation from their ancestor and they mate together, and this is first cousins, and when you draw your pedigree, first cousins, or if you have closely related individuals, you always use double lines, right? And then you get an offspring with it. So this one mutation was passed down, and then this offspring has the mutation. Okay. This in humans is called consanguinity. Right? This is the coming together of blood. Right? Consanguinity. And so if we look at some examples of consanguinity, right? This is Cleopatra. Okay. So the royal families of the past had a misconception that it was the royal blood that determined how great they were, right? And so they like to keep it within the family. And so you get to see lots of people marrying one another, right, and having children. So brothers and sisters, uncles and nieces, right? And so then you get a lot of abnormal mutations being present in the potential offspring, okay? Now you might think this is terrible, but this is actually what we do with inbreeding our animals, right? So if you think about dog and dog breeds, and you think about all the defects that they accumulate, like hip dysplasia, you know, early cataracts, they have all kinds of horrible health problems because we've inbred them, right? Maybe not to this extreme, but horses too. We've done a lot of inbreeding with horses as well. So I'm gonna, very last thing is there questions about that? Very last thing is I'm gonna show you a video that talks about the math, okay? So you don't need to fully understand this math, but the math talks about how we are all related, right? So when we talk about inbreeding and you go, ooh, right? You have to realize that mathematically we all share ancestors more recent than you probably would predict, okay? So.
Hey, Meiosis, Michael here, and you guys are my family. Scientifically speaking, geneticists tell us that every single person on Earth is at the least 50th cousins with everybody else on Earth. It kind of has to be that way. Think of it like this. You have 2 biological parents and 4 biological grandparents and 8 biological great-grandparents. It keeps doubling. Well, if you follow your family tree all the way back to the time of the ancient Romans, 64 generations ago, that tree would need 1 quintillion separate individuals, which is a number that is larger than the number of humans who have ever existed, ever. So what does that mean? Well, I think Bill Bryson put it best when he said, you couldn't be here without a little incest. Actually, quite a lot of incest. Nothing ridiculous, mind you, but if you go far enough back in your family tree, you're going to find that quite frequently, people who were distantly related had kids. In fact, if you are currently dating or married to somebody who is from your own country and is of your own ethnicity, there's a 1 in 5 chance that the 2 of you share a common family member fewer than 10 generations ago. Furthermore, because our society is becoming more and more mobile, our most recent common ancestor is becoming more recent. Mathematical models are now showing us that for everybody, every human on Earth right now, our most recent common ancestor probably lived only 2,000 to 4,000 years ago. A recent study, it's a great one, you should read it, found that there was a person alive as early as 300 BC who is related to every single person alive on Earth today. Their study also found that that person most likely lived in Taiwan, near a busy port city, so their descendants could quickly spread around the world. Now, all that said, you only receive half of your genetic information from your mother and half from your father. And as you go up your tree, it's quite possible that distant ancestors had literally no contribution to your genome. So, who are you? I've said this before, but because bacteria are so much smaller than the average human body cell, your body contains more bacteria than it contains its own cells. And forget about trying to remember every individual cell and bacteria in your body. They're constantly dying and being replaced. Even cells like brain cells, which seem to live for a very long time, still take in new nutrients and expel waste, meaning that within about every 5 years, every single atom that used to make up your body has been exchanged. Do you remember what you were like 5 years ago? Well, the atoms that make up your body, you sure don't. This is a little existentially scary, but it leads to a very good question. How many atoms are inside my body to begin with? How much is a billion atoms? Well, here's one way to think about this. If you were to count one number greater every second, it would take you 12 days to count to 1 million. But if you wanted to count to 1 billion, that would take you 32 years. So 1 billion is a very large number, but not when it comes to atoms. Atoms are so inconceivably small that even if you had 1 billion, say, sodium atoms, their entire weight would only be 38 femtograms. Each individual cell in your body, too small to be even seen by the eye, contains 100 times more atoms than there are stars in the Milky Way galaxy. Multiply that by the number of cells that make up what we call our cells, and each human body contains more atoms than there are stars in the known universe. Numbers that large lead to some pretty mind-blowing truths. For instance, this one mentioned by Bill Bryson. Because our bodies contain so many atoms, and because we so vigorously recycle them, statistically speaking, about 1 billion of the atoms inside your body used to belong to Shakespeare. 1 billion of your atoms used to belong to Beethoven. 1 billion of the atoms in your body right now, as you sit and watch this, used to be inside the body of Buddha. You can also think of it this way. 1,000 years from now, when humans first land on a planet outside of our solar system, that first person to step onto the surface of the alien planet, their body will contain 1 billion atoms that are in you right now. And as always, thanks for watching.
Okay. Okay, so I am going to record a short finished lecture on the Mendelian genetics. So you want to watch that hopefully before Tuesday's lab. Okay. We have lab Tuesday. No lab on Tuesday?
I was looking at the calendar, you didn't say that.
I think we do have lab. Yep. So lab on Tuesday. So make sure that you study for the quiz next Thursday and that you do your homework by next Thursday as well.